Okay, students, welcome back. This is part 2 of chapter 16. We're trying to figure out how DNA replicates. In part 1, we sorted out the structure of DNA, three-dimensional shape. Part 2 is all about how DNA replicates, and let's check it out. Nothing else exciting happening. So let's figure out DNA replication. So remember, DNA is double-stranded. It's two strands run in anti-parallel direction to each other. And DNA replication has to be accurate, right? Accurate, because you don't want to make a mistake in your genetic material. You don't want to pass along a mutation. So one of the things about DNA replication is it has to be very accurate. A second thing is it has to be kind of speedy. Think of human cells. Each human cell has 46 molecules of DNA, and on those molecules are roughly 3 billion base pairs. That base pair means a nucleotide, a pair of nucleotides. That's a lot of DNA that needs to be replicated if a cell wants to divide, right? We're thinking of a cell going through mitosis or going through meiosis. Before that cell can divide, it has to replicate all of its DNA. Has to do it quickly, has to do it accurately. So how is this achieved? First of all, when we look at replication, we say, where does it start? Where does DNA replicate? Well, in eukaryotes, like our cells, the DNA replication starts in many different spots along the way. Along the length of eukaryotic DNA are many regions where replication begins. These regions are called origin sites. Oops, I need to get myself organized here. Let's do red. These regions are called origin sites. Okay? Let's draw a picture of this in a second here. In prokaryotes, there's only one origin of replication. Remember, prokaryotes are much smaller. Their DNA is circular. Okay, they have one circular chromosome that's double-stranded DNA. And then they might have one spot somewhere on this circular chromosome. That's where DNA replication is going to start. But let's look at eukaryotes a second. So with eukaryotes, we have, a very long double-stranded molecule of DNA. If you want to put in the 5' to 3' and 3' to 5' designations, let's say this is one molecule of DNA. At different places along this molecule will be an origin site, a specific sequence of nucleotides where replication starts. Let's just say there are 3 of them in our molecule of DNA, but there would be many more. And at that replication site, DNA replication is going to go in both directions. So you're going to start replicating both directions from that origin site. Notice how fast that's going to be then, because we're replicating, starting at different parts along the chromosome, and we're replicating both directions. So what happens then, it goes something like this. If you've got at a replication site, you have the double-stranded DNA molecule kind of unwind from itself and form a bubble. We call it a replication bubble, actually. So at the origin site, the double-stranded DNA starts to unwind from itself, and then replication begins in both directions, and a new molecule of DNA starts growing, starts growing this way. So replication is going toward the left there at this fork. We call that little spot a fork, a DNA replication fork. And DNA replication starts going the other direction as well. So DNA is being replicated both directions at the fork, and pretty soon you get DNA replicating both directions, along each fork. And so we get rapid replication of DNA. And pretty soon we end up with 2 molecules of DNA that are each made up of one old strand, the red strand, and one new strand of DNA. Now all we have to do is sort out how this happens. Here's another picture of the origins of replication in eukaryotes, and you see it's pretty much what I drew as well, but you can see that the language that we'll use sometimes is a replication bubble and a replication fork. That's like the kind of juncture, right where replication is happening. Okay. All right. So how is replication achieved? That's the question. So let's see what I have. Okay. I'm just trying to figure out what I have in my notes. The first thing that has to happen is there is a protein called an initiator protein that finds the origin site and binds to it and kind of calls in all of the replication machinery. Hey, come here. This is where replication is going to happen, all right? Because we're going to need some enzymes. We're going to need some help to replicate DNA. We're going to need a lot of, and you can see, look at all the enzymes that we're seeing here. I'm going to change my color, I think, to, it's not letting me, to like a blue. Okay. After the initiator protein shows up at the origin site, the next, there are a couple other things that happen. One of them is that helicases then unwind the double-stranded DNA. So helicases unwind the double-stranded DNA at the origin site. We have to open it up so that it can be replicated, right? And once the helicase unwinds the DNA just wants to fold back up on itself. To hold the double-stranded DNA, to hold the DNA in an unwound position, there are things called SSB proteins, single-stranded binding proteins, that keeps the double-stranded DNA apart, right? So that keeps the SSBs, or single-stranded binding proteins, bind to the DNA and keep double-stranded DNA unwound. We need to have it unwound in order to replicate it, okay? That keeps the double-stranded, that's what DS stands for there, DNA unwound. So here's a picture, and we'll come back to these other things, but here's a picture of a piece of DNA and an origin site. So here's our origin site. Oops, I'm trying to draw it on here, and it's not letting me use the top part of our, like here is the origin site. And at this location, we've got a bubble. Notice that the double-stranded DNA is running 5' all the way over to 3', and the opposite or other strand is running anti-parallel to it. On one of these strands, I've put a couple of nucleotides, ATT, CCG. Okay. And what we've got so far is a slightly unwound double-stranded DNA, and I'm going to put into place a helicase enzyme. You should label it helicase. I'm just going to label it with an H because I can't write all those words out. The helicase enzyme has bound and now is unwinding that double-stranded DNA, so we're getting a big bubble. Then there are single-stranded binding proteins. Maybe do these in a different color. I didn't. That bind to the single-stranded DNA molecule. I'm going to put a couple of them in here. It's not liking me at all. I didn't do it. I don't know why. There we go. There we go. Single-stranded binding proteins bind to the single strands of DNA and hold them unwound so that we can replicate the DNA. All right. As we're unwound, and then down here we've got a bigger picture of the bubble, right? So I could put in, these A, T, T, C, C, G, if we wanted to. Okay. Now, what happens when we unwind DNA at this location is that we start to uncoil it. What happens to the DNA kind of upstream from this bubble? We're unwinding it here, and what happens downstream? What happens like over here, or what happens over here? As you unwind one part of double-stranded DNA, what do you think happens to the parts upstream? They get hyperwound. They get overwound. It's a little bit like, when you're pulling out your Christmas lights and you start to pull apart a couple of Christmas lights because you haven't put them away very well. This happened to my dad every year when I was a kid, and he'd get really angry. And he'd start pulling at the Christmas tree lights, right, to unwind them, to separate them. He'd be like, yeah, this is working pretty well for a while. You can kind of unwind them for a while, but what happens upstream? Well, you got a disaster, right? The rest of the chain gets all messed up. That's what happens with DNA too, except there are enzymes called topoisomerases. Add a topoisomerase, a big guy. We're going to call that the topoisomerase. Topoisomerases will bind to the double-stranded DNA upstream of our replication bubble, and they will unwind. Well, basically, they will cut the DNA, unwind it, and put it back together again. They will, what do I have for my language here? I think I wrote something like this. Topoisomerases relieve the twisting DNA molecule by clipping and rejoining DNA. So if you want to put that in here, topoisomerase enzymes relieve the twisting DNA molecule by clipping and unwinding or rejoining the DNA strands. All right. Now we just are ready to go. All we have to do is build DNA, right? We've got the DNA the unwound. We've got it held in its unwound position. We've dealt with some twisting DNA upstream, and we're relieving that. Now we just need to build DNA, but we're not quite ready to, because the enzyme that builds new DNA can't start from scratch. So if I wanted to build new DNA, and I just wanted to just start building it, maybe I need to do something here. Maybe I need to write in, hold it here, maybe I need to draw in the, why is this not liking me? I really want to change the color. I really, really, really want to change the color, and it's not liking that. Oh, well, that worked. Okay, so I don't know if I wanted to change it anyway, but maybe I want to write in the complementary strand of DNA that matches ATT-CCG, but I'm going to put it up here, okay, just so we know, TAAGGC, right? Okay, so if you want to build new DNA, it'd be easy, right? You would just say, oh, I see a C here, I'm going to add a G. I see a G here, I'm going to add a C. I see a G here, I'm going to add a C. I see an A, I'm going to add a T. I see a T, A, I'm going to add a T. Okay? It'd be pretty easy to build new DNA, so you'd think that an enzyme should be able to do this right off the bat. DNA polymerase that builds DNA can't start building new DNA from scratch. So at every single origin site, there is what we call a primer that's built by an enzyme called primase. Let's go back up, we'll fill in this blank. Primase is an enzyme that synthesizes RNA. Oh, what? Primase is an enzyme that synthesizes a short RNA chain called a primer. So write in your notes, a short RNA chain called a primer, and the new DNA strand will grow from this primer. What on earth does that mean? Well, here's what it means. I'm going to draw the primer in red. There is an enzyme called primase, and I'm going to build it in, make it like a little blob. Primase that has to build a short primer of RNA molecules. Oh, RNA molecules. And it builds these RNA molecules in only one direction, in the 5' to 3' direction. So it lays down a short RNA sequence called a primer. The primer complements the DNA. So when there's a C, it puts in a G. When there's a G, it puts in a C. When there's a C, what was that? A G, it puts in a C. When there's an A, it puts in a T. Oh, wait a minute. It doesn't put in T's. It's RNA. It would put in a U. Okay. A short, maybe 10, 12 nucleotides long, a short primer made of RNA has to be laid down before we can build DNA. This seems really strange, but that's what happens. And I had to make this clear that the primer has to be built in one direction. Primase only builds new DNA in the 5' to 3' direction. So it builds the G, the C, the C, the U, the A, etc. and it only works in one direction. Starts on the 5' end and builds on the 3' end, or adds to the 3' end. So at every single origin site, a primer has to be laid down before new DNA can be made. It seems really awkward, but that's what happens. Let's draw in the blanks here. See if we can fill these in. The upstream of our replication bubble, we've got this molecule unwinding and relieving twist in DNA. That's topoisomerase. You can finish that. Topoisomerase. The helicase unwinds the parent DNA. The single-stranded binding proteins hold the parent DNA unwound, and primase, an enzyme here that's shown in red, builds a primer. And notice that our primer is being built in the 5' to 3' direction on that strand. Now we're finally ready to build new DNA. And what we use to build new DNA are DNA polymerases. They polymerize DNA. They put nucleotides together. They catalyze the synthesis of DNA. Yay, we're going to make DNA by adding nucleotides to a growing chain of nucleotides. See what I mean? They can't grow from scratch. They have to add nucleotides to a growing chain. The growing chain, folks, is this little primer that's already laid down. Let's take a look at a picture of DNA polymerase in action. And then we have a couple of things that are starred here. This means they must be really important, all right? Turns out, by the way, there are different kinds of DNA polymerases down here. For our purposes, let's just say DNA polymerase, a type of DNA polymerase, right? And DNA polymerase builds new DNA pretty quickly, much more quickly in bacteria, but still it can lay down 50 nucleotides a second in eukaryotes. So here's a picture of, let's orient you a minute, a little piece of DNA. And in this little piece of DNA, we're adding a new nucleotide because we're building DNA in this picture. What we have, first of all, is a template strand. That's the original strand that we've unwound, and we're using it as a template to build a new strand of DNA. Also notice that these are the template strand and the new strand run in different directions. They're anti-parallel to each other. The bases pair up so that A pairs with T and C pairs with G. And it looks like we need to add a base here. We're trying to add the next nucleotide, right? And we're adding it to the 3' end of a growing chain. So here's our chain. We're adding it down here at the 3' end, okay? It's the only place that DNA polymerase can add. It can't add up here. We can't put something new up here, okay? Can't do it. It can only add nucleotides at the 3' end of a growing chain. So what DNA polymerase does, and here's our enzyme here, this is like a reaction shown in the picture, right? DNA polymerase finds and inserts the appropriate nucleotide. And so what it does is if it sees an A, it goes, oh, I need to add a T nucleotide. It adds a T nucleotide to this 3' end of a growing chain. And as it adds it, it really adds, it's not quite a nucleotide, it's called a nucleoside triphosphate because it has 3 phosphates sticking out there. It's not going to keep all those phosphates, it's only going to keep one. 2 of the phosphates are removed, as you can see, and they're kind of a source of energy. So what it does is it adds the nucleotide, it makes this phosphodiester bond, or that bond right there, happen, and now it's ready to add the next nucleotide. What would be the next nucleotide that comes in here? Well, the template strand has a C. What should I add? Well, I'm going to add a nucleotide with a G, right? And it can keep building, and it builds the new DNA 5' to 3' on the new strand. So it only can build in this direction. It cannot add things up at the top end. only build 5, we always say it builds 5' to 3'. Okay, so that's the second important point. The first important point is you have to have a starting point. You have to have nucleotides already in place to add to. That's the primer. You also can only build new nucleotides in a 5' to 3' direction. You can only keep adding nucleotides on the 3' end. Okay, that's why it grows in the 5' to 3' direction. That's how DNA grows. Okay. All right. And so if we were building on this molecule here, and we had, let's just put these in, T, A, E, G, C. And down here you have A, T, T, C, C, G, right? If we have this molecule here of DNA that's been unwound at its origin site, remember it's running 5' to 3' on the top strand and 3' to 5' on the bottom strand. We would first need to build an RNA primer. Okay, I'll build that in red. The RNA primer lays down in a 5' to 3' direction. So on the upper strand here, the RNA primer builds a short sequence of RNA, in the 5, this is 5' to 3' direction. It builds and it matches up with these little guys. Now DNA polymerase, let's do a different color for DNA polymerase. DNA polymerase can come along and build new nucleotides, DNA this time, that build in the 5' to 3' direction. So again, you can put in your 5' and 3', and they would add nucleotides that complement, the A, the T, the A, and the T. So we'd put in a T, and an A, and we would keep going. And as this replication fork unwinds, this DNA polymerase, let's call this guy DNA polymerase, can continuously replicate the DNA on this strand. It keeps on moving. As the unwinding happens, the DNA is replicated and more DNA is replicated, etc. Yay, DNA polymerase is building new DNA. We're making a new strand. Now, let's see if I'm here. Yeah. The DNA polymerase can replicate in the 5' to 3' direction only. So what that means is at any replication fork, one of the templates or one of the new strands of DNA that's being built is being built in a continuous fashion. We needed a primer, but once we put the primer in place, and I'm going to write down that that's the primer there. Once we put that primer in place, we could build DNA in a continuous way, 5' to 3' as we opened up our replication fork. Right? No problem. The other template strand of DNA is running in the opposite direction. And at this replication fork, as we open it up, DNA polymerase cannot simply, easily build DNA continuously along this fork. It cannot continuously lay down nucleotides going this direction because it can't build DNA in the 3' to 5' direction. So let's look at some pictures and I'll try to draw this out again. Okay, this is kind of a strange picture, but it's a picture of a little part of a replication bubble. So here's our origin of replication up here, and here's our bubble. Overall, we've got DNA being replicated in both directions from that bubble. And we're looking at what we would call the leading strand of DNA replication. So far, that's all we've looked at, this little box here, all right? This little part of a replication bubble. We laid down a primer. That's what's in red. Then then DNA polymerase came along, and DNA polymerase then builds new DNA, and it builds it all in the 5' to 3' direction, and it can build DNA continuously on the leading strand of DNA replication. So we see this beautiful new strand of DNA being built in the 5' to 3' direction. The problem is what's going to happen over here on the lagging strand, okay? What's going to happen on this side of the molecule of DNA? That's called the lagging strand. Let's take a look. What's happening at the lagging strand? That really is our question. What is going on at the lagging strand? Okay, let's fill these in and then we'll take a look. So in order for any DNA replication to happen, in order to start replication, we always need a primer. So a primer must be in place. We have to build a primer first. That's made up of RNA. The lagging strand is then synthesized in short pieces or fragments. These short pieces are called Okazaki fragments. Okazaki fragments. They're named after the person who discovered them, all right? So we're going to see that on the lagging strand, there's some problems. You can't build DNA continuously at the replication fork. You have to build it in little pieces. All the pieces are being built 5' to 3'. Eventually, though, you have to join together all the little pieces into one continuous molecule of DNA. All right, so we need to take a look at the lagging strand. This is what we're really looking at down here. We're looking at this replication fork. Okay, we've already studied, this part of it, the leading strand. We added a primer, and then we build new DNA in a 5' to 3' direction. Pretty easy. But over here, on the lagging strand, as you're opening up your replication fork, you have to lay down a primer, and the number 1 there means that's the first little piece that's built. A primer in red is laid down. Then you have to build DNA in this direction because you can only build DNA 5' to 3'. Then you have to unwind the replication fork a little more and lay down a second primer that's in red by number 2 and build more DNA in the 5' to 3' direction. Notice that the template strand runs the opposite direction. And then you have to do it again and again and again until you get you're building DNA in little chunks. Notice also that at the other side of the replication fork, just for fun, to make things more complex, what was the leading strand now becomes the lagging strand because you have to build DNA in the 5' to 3' direction. You have to build short fragments of DNA. Over on this side of the replication bubble or the replication fork, after you've laid down a primer here, you can build continuously 5' to 3'. So at a single replication bubble, each new strand being built on one side of the fork is called the leading strand, and on the other side is called the lagging strand. Crazy, but true. Think about how this is happening inside your cells. Every time your cell has to build a new cell, it has to replicate all of its DNA, all 46 chromosomes of its DNA in this crazy manner and not make any mistakes. And so let's draw this in. So this is going to be my leading strand, and I know that based on the direction of my molecule here. But what happens is after a primer is laid down, I better lay down a red primer. After a little primer is laid down, 5' to 3', then DNA polymerase can come along and build new DNA continuously in the 5' to 3' direction. I'm going to put my polymerase there, and it builds boom, 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 boom. It's building new DNA continuously. That's the leading strand. The lagging strand is much more complicated, right? That says lagging. It kind of says lagging. So as we first open up the replication fork, DNA polymerase is going to lay down, RNA polymerase is going to lay down a primer in the 5' to 3' direction. Then DNA polymerase can come along and build new DNA. continuing on the primer and building new DNA, okay? The replication fork opens up a little more, and we have to lay down another primer, 5' to 3', okay? Then DNA polymerase can come along and build new DNA, 5' to 3'. And then we open up the replication fork a little more, and we're going to have to lay down a new primer, 5' to 3'. And then DNA polymerase can come along and build new DNA, 5' to 3'. So we get little fragments of DNA being built, maybe 100, 200 nucleotides long. That's the lagging strand. We're going to have some problems. We have to eventually have a complete molecule of DNA. We're going to have to get rid of anything that's red in our drawing. So we're going to have to get rid of all of the primers, and we're going to have to connect all of these little pieces of DNA, right? Here is a picture of the replication of the lagging strand, and it's awkward. It's an awkward drawing. That's all I can say. I think there are some animations on our Pearson site that would be worth doing. But let's just run through all of my words here first, right? So the first thing that has to happen is helicase unwinds the DNA molecule. Primer lays down a short new RNA sequence called a primase lays this down, called a primer. It runs in the 5' to 3' direction, and then DNA polymerase, shown here in orange, makes the first Okazaki fragment, builds a little fragment here, and then the polymerase detaches, DNA polymerase detaches, and now we need primase again to make a second primer. Here's our 5' to 3' primer, and then, here it is, right here, our second one being laid down, and then DNA polymerase comes along and builds new DNA, and then it happens again and again and again as we open up the replication fork at the lagging strand. Eventually, we have to get rid of all of the primers and connect all of the DNA together. And this note here says a different DNA polymerase removes the RNA primer. Oh, so we have to get rid of it. A different DNA polymerase, shown here in yellow, removes the primer and replaces it now with DNA nucleotides. It can build from its other DNA, and it kicks out the RNA, puts in new DNA. So another type of DNA polymerase, don't worry about the type. But finally, we're left with a whole bunch of pieces of DNA, but they're not connected to each other. There are little breaks or gaps wherever there had been a primer, right? And so there is another enzyme yet called ligase, and that's shown in green here, that has to ligate or glue together the newly synthesized DNA fragments. So these are the things that we just mentioned. Primers must be removed. You got to get rid of the RNA and put new DNA in its place. After the primers are built and used, then they need to be removed. DNA polymerase removes them and replaces the primer with DNA. But it cannot join the final nucleotide to the already in place nucleotides. They can't make that final connection. And then there's an enzyme called DNA ligase that joins them together. Okay? Yes. I like this summary slide. It's sort of static, like our video has been, but it kind of helps you understand what's happening on the lagging strand in particular. It recognizes that you have to recognize that this is all happening continuously. One of the things I wanted you to notice is it names DNA polymerase as DNA polymerase 3 and DNA polymerase 1. We're not going to worry about the Roman numerals here. We're just going to call them a type of DNA polymerase, right? There's also a ton of DNA replication videos. This one is what I recommend, but there are lots of them to look at. So check them out. I think they will help you enormously. This is one of these things that you need to actually do to understand DNA replication. Let's see if we can answer all of these questions. I guess we'll do them in order, but I'm looking at a little section of a replication bubble. Also, it says look at the Mastering Biology animation here. That also might be good. And I think that we've already unwound the DNA. I'm guessing that the first blank there would be single-stranded binding proteins. The second blank would probably be the molecule that's unwinding the DNA. That would be helicase. Helicase does the job of unwinding the double-stranded DNA. It looks like this number 3 is DNA polymerase because it's building a strand of DNA. So this would be DNA polymerase. I'm just going to write DNA pol. Number 4. It looks like number 4 is an enzyme building a primer. So that enzyme is primase. Number 5. Looks like we're talking about the newly synthesized DNA that is being synthesized in a continuous way. So that would be the leading strand of DNA. Number 6, it looks like that's a chunk of newly synthesized DNA, so I would call that the lagging strand, or it could be called an Okazaki fragment. Oh, maybe we should write Okazaki fragment, probably. I'm not going to rewrite that. Number 7, it looks like we're dealing with another DNA polymerase here that's building our DNA. And number 8, we have an enzyme that's ligating or gluing together the newly synthesized DNA fragments. That has to be DNA ligase. Look how much it takes to build new DNA. Look at all the molecules involved that we've been talking about. Building new DNA is complicated. That's the end of video 2, and in video 3 we'll continue with this and look at just a couple other topics that are found in chapter 16. That's tough. If I were you, I recommend definitely looking at animations of DNA replication, trying to draw it out, because that's going to help you enormously in understanding it. I think that's it for now. We'll talk to you later.